All right, so tonight we're going to move on with cellular reproduction. So section 8.1 is giving us a bit of an overview. So the first thing that they want us to know is what is the purpose of cellular reproduction? So there are three major purposes. One of those is growth. So you start off as a single cell. This single cell is called a zygote. You will undergo cellular reproduction, and over time, this will allow you to become a multicellular organism. So, when the sperm from your mom meets the sperm from your dad meets the egg from your mom, this produces a single cell. That single cell will then undergo reproduction. One cell will become two, two will become four, four will become eight. We will keep growing until we were multicellular. So overall, this just allows for growth. It also allows for replacement. Over time, tissues become worn out. So worn out tissues or damaged tissues, these need to be replaced. So you will undergo cellular reproduction in order to replace them. Your blood cells will be replaced every 2 to 3 months. Your entire skeletal system will replace itself every 5 to 10 years. You're constantly losing cells off of your integumentary system, your skin. These are being replaced at a daily rate. And then lastly, the third purpose of cellular reproduction is this is how you make new organisms if you are single-celled. This is a process known as binary fission. So remember, we learned that bacteria and archaea and some protists are single-celled organisms. If you want to make the next generation, you will undergo binary fission so that you can make more of your group. So we're multicellular. We do not do binary fission. We will produce a single-celled zygote that will then undergo cellular reproduction. But if you are single-celled, this is the only way you can make more of your species. All right, what's the next thing? Understand the relationship of sister chromatids to chromosomes. So they did this a little bit out of order in your book, and they actually put the figure for this in section 8.2. So when you're doing this particular learning outcome, you're going to be wanting to look at figure 8.4 in the book. So if you recall, when we were talking about the cell structure in the first day of class, we have DNA. And that DNA is set up as units of chromosomes. So here's a segment of, let's say, chromosome 1. And you have 2 copies of every chromosome. So this is like A1, 1A, and 1B. You also have 2A and 2B. All told, actually, you have a total of 46 chromosomes. Each of them is a linear segment. So if we were to draw them all out, you'd have a whole bunch of these linear segments. So right now we've shown 6 of these chromosomes. So if we were to draw them all out, we'd get to the end and we'd have 45A, 45B, 46A, 46B. But you would have a total of 46 of these linear segments. Now, for part of your life, these chromosomes, so this here is 1 chromosome. But when you have a single unit of DNA, it's also called a chromatid. So at certain points in your life, 1 chromosome is going to equal 1 chromatid. At a certain point, though, you're going to want to make more of your cells, and in order to do that, you're going to have to double the amount of DNA. when we double the amount of DNA, we don't double the number of chromosomes, we double the number of chromatids. So during what is called DNA replication, we will double every one of those 46 chromosomes. So when we're done, you will have 1A is now 2 units. 1B is now 2 units. 2A is now 2 units. 2B is 2 units. 3A, 3B, dot, 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 all the way to the end. Here's 45A, here's 45B, here's 46A, here's 46B. Now, one chromosome has 2 segments. This is still one chromosome. But now, because it's made up of 2 linear units, it is now composed of 2 chromatids, or what we call sister chromatids. So during DNA replication, we will double the amount of DNA. One segment became 2. One segment became 2. One segment became 2. One segment became 2. You had 46 segments, 46 chromosomes with 46 chromatodes total. When you're done, you'll still have 46 chromosomes, but now you'll have 92 chromatids. So DNA replication means we will double the amount of DNA. And so they show this for just one chromosome in figure 8.4. So they show you a single chromosome. So this is one chromosome composed of one chromatid, one unit. After DNA replication, so here's our DNA replication. We will make a copy of this, just like if you went to the photocopier and then stapled it to itself. We will make another segment of DNA. Now we still have one chromosome composed of 2 chromatids. Or the other word for 2 chromatids are sister chromatids. And then another term that's important here is these 2 chromatids are tied to each other by some proteins, and this is called the centromere. So this is an important relationship. When we get to the next section and we start talking about how we go through the specifics, if you want to make a new cell, you've got to give that new cell a copy of the instructions. So you'll have to go through a process of DNA replication, where you'll take a set of instructions and you will double it. Both are called chromosomes. One is called a chromosome unreplicated. So you might see people say this, an unreplicated chromosome, because it's only one segment. And these are often called replicated chromosomes, because now we have an identical copy of that. One will stay in the cell that is going to divide, and the other will go to the new cell so that it can be a fully functioning cell. If you don't have DNA, you can't compose the new cell. All right, so that's the relationship between chromosomes and chromatids. Chromosomes stay constant. Chromatids double. That's one of the challenges of this section, is there's going to be a lot of words that sound alike. And then also, we need to know about 3 chromatin will condense to become a chromosome. So chromatin is when DNA is in an uncoiled linear fashion. So you see here, my chromatin is very stringy. If something condenses, it becomes thicker. When we condense, we take this same thing and we ball it up so that it's tightly bound. And you notice this is now coiled and smaller. And the reason that we need to do this is when we go to transfer that DNA to another cell, if you have all these strings, they're just going to get tied up in a knot. It's going to be very hard to get half to this cell and half to the next cell. But if you condense them, it's much easier to take this small package, tie it up really nice and tight, and then it's easier to give off to the next cell. In order to go from being chromatin to a chromosome, when we have DNA in a cell, it actually is going to tie itself around some proteins called histones. And there's usually 4 of them together. So here's our histones, and this DNA can be unorganized and all over the place. So these are the histones, and then here's that linear DNA, and it is wrapped around these histones. When we go to condense these chromosomes, these histones will begin to line up and they will become units called nucleosomes. So this here, the proteins plus the DNA, this is called a nucleosome. So right now they're just sort of strung out. But the nucleosomes are now going to organize themselves. So we start off with just regular chromatin wrapped around histones. They will then begin to form a zigzag pattern. So this will become zigzag chromatin. And when they do that, that means that these nucleosomes will line up in a pattern that's going to go back and forth. It's going to zig and zag. So you'll have one on this side and then one on this side. You notice now we have this zigzag pattern. The DNA is still wound around it. So we'll put our DNA in here. So here it is round around this one, and then round around this one, and then wound around this one. But we have a zigzaggy pattern. Next, this chromatin in the zigzag pattern is going to start to loop. And so then we will get looping chromatin. So now all these zigzags, and I'm just going to draw this out as a blue line because it's hard to get it. All those zigzags are now going to be making these loopings. You notice it's starting to sort of compress itself. So we go from just regular strung out chromatin, they get into a zigzag pattern, then they get into a looping pattern, and then finally those loops will sit on each other, and they will become really condensed. So now all of a sudden these loops, they're going to be right next to each other. And you notice, look how big this was. Look how big that is. This is a big stringy mess. Here we have our chromosome, which is condensed chromatin. So the reason that we have chromatin condensed to become a chromosome so that it's easier to pass to a new cell. It's much easier to have this little package than it has all these stringy packages when we're trying to divide and become a new cell.